Cool. And I just started recording.
So, hi everyone. My name is Kate. I'm a graduate student here at UCSC studying bioinformatics, and I'm doing lab research specifically focused on trying to reverse engineer how the brain works using these models called cortical organoids, which are in vitro balls of cells that we can grow and study from stem cells. So today I just wanted to have a general overview for you guys about neuroscience, specifically how does the brain work, how do neurons communicate with each other, and potentially how can that help us interface with the brain, which is something we're all interested about. So the first question is, how do we tie together computer in order to do brain-computer interfacing? How do we tie together computer and neural computation? And there are some unifying principles which we can go off of. For example, both the brain and computers have some kind of memory storage. They both have some signal propagation or communication between different components, actually both of them are electrical, but whether it's neural, wet type signaling or in silico, those are differences, but there's both propagation and communication of signals. And there's operations that are done on those signals. For example, a computer can do multiplication or addition using the signals it has stored in memory. And the brain similarly can have signals pass through neurons, and those neurons would modify the signal that they pass on to other neurons they're connected to. And we can think about computation in both the brain and computers as a part of just general information theory, where you have some kind of information that gets manipulated and acted upon or stored, and that's similar in both systems, independent of what materials they're made of. And so to create a brain-computer interface that tries to connect these unifying principles, we first have to have physical interfaces, such as electrodes. Right now, actually, a really hot field is optogenetics, where you try to stimulate neurons with light, and also neurons can be genetically modified to emit light when they're excited. So either it's electrical sensing or imaging light. How can we create an interface, physical interface, between neurons and some computer? And then the second challenge is how, after we've been able to record neurons and get signals on paper, how do we understand what the signals mean? And that's also a really important part of being able to create good brain-computer interfaces, is being able to decode what signals mean certain things, and how can the computer respond appropriately? Or in general, how can we understand what's happening at the core of neural communication? And kind of the third step of our engineering challenge is, now that we have interfaces and we have some understanding of the neural code, can we create adaptive software systems, like maybe neural networks or some combination of kind of adaptive programming to have a very organic and easy interface for a person to be able to use that brain-computer interfaces to do practical things in the world. And so this lecture today will mostly touch upon physical interfaces, as well as understanding the brain and neural signaling. And later on, we can talk about software systems in a different meeting or after this meeting. It's just a fun topic that's still up in the air, and it's definitely a really cool research area. It's how to create software. So the first step is, how do we measure neural signals? And we can do this by sensing the activity of the neurons in the brain. This is over here a typical neural spike that you might measure. And a neuron usually has a resting potential. And after some time, it gets excited. So there's a peak in voltage. And afterwards, it tries to restore to the baseline. It overshoots a little bit and goes back to normal. And we can go into the details of why and how this curve happens. But firstly, we can measure neural activity in many different ways. The spike that you saw before here would likely be measured by a very small probe that's close to the neuron, such as this electrode array. But we can see more global and overall activity using other types of electrodes, such as EEG or MRI or fMRI. And so I really like this video by Neuralink, which at the beginning explains how neurons function. Can you guys hear the audio?
Enough of the right kind of neurotransmitter.
Rohan, can you hear audio? Just wondering.
A chain reaction is triggered that causes an action potential to fire, and the neuron to in turn relay messages to its own downstream synapses. Action potentials produce an electric field that spreads from the neuron and can be detected by placing electrodes nearby, allowing recording of the information represented by a neuron.
Yeah. So you guys were able to see the cells and electron and how we're able to capture those signals a little bit. In general, we have a probe that's sitting in some location in the brain. This is specifically for neural scale. We have a probe that's sitting in the brain, and we have some reference point, which we're measuring against. And that way we can get some voltage difference, which means that there's certain activity in this area that is different from a baseline ground signal that we're comparing to. And we can get these kinds of neural spikes that you've seen before. These are flipped, actually, because we're measuring with respect to inside versus outside the cell. If a probe is inside the cell, the signal will be flipped. And if the probe is outside of the cell, it'll be this downwards curve instead of that upwards curve that you were seeing earlier here. It's just a matter of reference and what you're measuring with respect to. So there are all kinds of electrode probes. These are microelectrode arrays. They're flat and they're quite small. They can be 2D flat, or they can stick out a little bit from the surface, like this Utah array, for example. And then we have silicon probes, which can be on these needle-like structures that we call shanks. And each one of these pads is an electrode that we can measure from. There are also electrode probes that you can make out of carbon fibers. These are in development and they're quite cool. They're also flexible and they're very small, which makes them a really good probe for getting into the brain in a very delicate way. So when you're inserting these probes, if they're really thick, you might actually pierce and puncture neurons. And also when a probe damages the brain, you have other supportive cells that are there to nurture and protect the brain coat these electrodes so that they insulate them and you're not able to record properly. So basically they're trying to reject the implant and because there's so many cells around the area, supportive cells, they will insulate and stop any kind of useful signal recording. So having small and flexible electrodes is important, not to have that kind of effect. We also have tetrodes, which are also quite small, And the cool thing about them is that they come in groups of 4. So you're able to pick up on a certain area with 4 very nearby electrodes, and you can get a little bit of a difference in your recording and be able to spatially orient better, just because one electrode here might sense a neuron here, and it'll be closer to that neuron as opposed to this electrode, so you can kind of get a spatial characteristic. These are just some more examples of electrodes, the MEA, tetrodes that you see here. They're just little tiny floppy wires, and silicon probes, which have little electrodes here. These are just some more pictures of silicon probes. They can come with light stimulation as well. And these are the results that you might get from a neural recording. This is that same neural spike that you've seen earlier, flipped upside down, because we're measuring on the outside of a neuron as opposed to inside. And this is something called a spike raster. And it's just a timing diagram of where we computationally identified that neurons fire. So on the y-axis here, you have units, or in other words, things we think are neurons based on our recording. And on the time axis, we just have the time. And we can say that a neuron, hypothetical neuron 14 was firing this many times with this pattern, and hypothetical neuron 10 was firing less amount of times. And we can try to pick out correlations. For example, some neurons fire at the same time, so maybe they're relatively at the same time, very close. So maybe they're connected, and one neuron is stimulating another one, or maybe they're in some kind of network. So we can do some interesting analysis of this. And the way we get these signals and these timing diagrams is through an algorithm called spike sorting. This is just a general name for an umbrella of algorithms that try to decompose signals that you might get from an electrode into specific time points of neural firing. So we try to pick out where in the data we have neural spikes, and sometimes the neural spikes will be overlapped with each other, and we have to kind of deconstruct and pick out specific units through clustering. And so the general idea is to think of this as kind of the cocktail party problem, where you have many people in a room. You might have a band, you might have someone talking, you might have a police car in the back, and they're all at different distances from each other, and they all have characteristic waveforms. So different neurons might have slightly different waveforms, and we can use that to our advantage. So location and neural waveforms, differences in neural waveforms can be used to deconstruct voltage recording from the brain on many channels. So many channels are like multiple microphones in a room. And we can say that the man was close to the band and far away from the police vehicle. So this electrode that's closer to the police vehicle would have the signal be louder and the man and the band be less loud. And so by having different kind of compositions in these mixtures, we're able to decompose and separate out our sources. So kind of transitioning into a different kind of probe, which is EEG, or electroencephalogram. We have much larger electrodes, but the principle is still the same. They sit on some area of the scalp, in this case, not inside the brain, but they're able to record a voltage on the scalp with respect to some ground point, which is usually the ear, or sometimes it's, I think, back of the neck or some bony area. And each electrode can sense a voltage with respect to the ear, for example, and you would have differing signals based on the internal activity of the brain underneath the skull. And EEG, again, is recorded from the scalp. We can measure clusters of neurons, not specific individual neurons, but an area that's about the size of a quarter per electrode. And instead of capturing specific electrodes, neuron spikes, we're capturing more of ensemble activity of neurons, contributing all together into a larger waveform. And we can pick out rhythmic activity that happens due to neural oscillations, kind of oscillations between regions of the brain or specific neural circuits, clusters of neurons connected to each other. And that can be manifested in many different brain waves, like gamma, which is correlated to awareness, beta, which is correlated to alertness, alpha being relaxed. I think during meditation, for example, they're currently selling headsets that can do that can help you in meditation practice. That can tell you when you're not clearing your mind and relaxing. And those might be looking for alpha waves to make sure that you're in the right zone for the practice. Theta, tired, and delta is usually talked about in sleep studies. And here you can kind of see spatial resolution between something like a penetrating electrode and an EEG, which has a much larger spatial resolution, but the time resolution is still fairly close as compared to something like an MRI. And whenever you're recording electrodes, this applies to the invasive probes as well as the electrodes outside the brain. You start with some neurons, and those generate a signal which is picked up by electrodes, whether inside or outside the brain. And those signals usually need to be amplified because they're quite weak compared to the noise in the environment. And after amplification, you want to convert the signal to a digital form. Instead of being an analog, it becomes a binary number that can be stored in a computer. And with that data, we can start doing interesting things like controlling robots or maybe affecting something that happens in the computer, such as moving a mouse. And there's also a really cool field of neurofeedback, which is taking measurements of brain activity and then seeing the activity that you're getting. For example, just putting on a headset and knowing that you have gamma waves or beta waves, or that you have alpha waves, can trigger you to produce more alpha waves just by being able to visualize your brain recording. You can kind of start manipulating what happens inside of you and see the effect. And so now that we've reviewed some electrode recording techniques, we can talk a little bit about what happens in the brain and what can we understand is going on. And the following slides are taken from David Hostler's lecture in BME 230B. So all the credit goes to him for the slides below. So the skull is covered by a bone, and bone is usually where you would have something like a EEG electrode. And underneath the bone, we have the dura mater, which is kind of a sac that protects the brain. It's quite tough. And underneath that, we have some blood vessels and cushioning before we have the brain. And this is the brain underneath the skull and underneath the dura mater. And the most prominent feature that you can see here, which is covering all this entire area on the surface, is the cortex. And if you unwrap the cortex, it's about the size of a dinner napkin, and the thickness is about 2 to 4 millimeters. And we're able to pack so much area because the cortex is shriveled up and wrinkled into the skull, which is different from other animals. For example, if you look at a mouse brain, it has a lot less of these ridges or sulci or gyri compared to humans. And this is just a cross-section of what you see inside the brain. Of course, not all of the brain is the cortex. Cortex is just outside here. But this is the brain is split into the right and left hemisphere, and this is just the right hemisphere. And you can see that there are distinctly different areas that could potentially hint at different functions of the brain. And just in general, the brain evolved from the bottom. So we like to think of the bottom part here as the reptilian, the reptilian parts, like the hindbrain, and it has some clear evolutionary analogs with other tetrapods. And then we have the midbrain, which contains the limbic system, which is responsible for emotions. And that's specific to mammals, for example, monkeys and humans. And then on top, we have the cerebral cortex, which is not unique to humans, but is largely a human part of the brain, most developed in humans. And this part is responsible for higher level cognition and analytical reasoning and other sensory areas such as visual cortex here and motor cortex and sensory cortex. And these are just the reptilian parts of the brain that you would see here. They help with balance and just general sustenance of the body, like breathing or pumping blood, things that we don't consciously control or try to think about. And then on top, we have olfactory bulbs, which help us with smell, and the thalamus, which is like a signal relay inside the brain. It allows us to relay signals from different parts of the brain through it. And the hippocampus, which is connected to memory. And for example, the pituitary gland, I think, is connected to thought, or I'm probably getting this wrong. It's been a while since I've reviewed, but basically this would be the midbrain area. And then on top we have the cortex, which has different lobes. For example, the frontal lobe is the one responsible for analytical thinking and cognition that we attribute to humans largely, since humans have the largest frontal lobes. And then we have the parietal lobe, which is connected to sensory information. Occipital lobes, which is responsible for vision. And actually, if we put electrodes near or over the area of the occipital lobe, we can detect and try to pick out and correlate what the person is seeing with those signals, and there would be a fairly high correlation. And we have the temporal lobe here, which is responsible for formation of memories and time sequences. And it's interesting that the primary somatosensory cortex, which is along the ridge here, and the primary motor cortex are very close to each other. So there's a link between different functions of the brain and where they are positioned spatially. So this would be sensing information, and this would be acting. So this, for example, here you might sense a rough surface with your finger, and here you might be able to move your finger in certain areas, or ask your body to move the finger in certain areas. And the motor cortex and somatosensory cortex are quite interesting as well, in that they contain distorted body maps. So actually the area of the region can correspond to the amount of sensory information that you can get from a certain body part. For example, fingers have a very large dedicated area to them, so we can sense a lot more with our fingers than we can, for example, with our toes, potentially. So there's a little bit of a scaling. And a lot of the major brain regions that you've seen are developed in utero. So humans are interesting also in a sense that a lot of the brain has to develop inside. Actually, for all mammals, a lot of the brain has developed inside before birth. But for humans, later on, they spend a long time adapting to the environment and learning post-birth, which is why human children are quite vulnerable and need a lot more time for growth as opposed to deer or other animals that are just ready to act in the world right off. And so the brain develops in utero, and most of the neurons are already in place at 5 months. And there are many different kinds of neurons that are happening inside the brain, and they all have different functions and different types of processing modalities that they have. And neurons communicate between each other using synapse connections. That's one of the most prominent ways of connecting. And here we have presynaptic neuron, and in here we have the postsynaptic neuron. This is the synapse, and the neuron before that's passing on a signal is the presynaptic, and the neuron that's receiving is the postsynaptic. And here we have an axon from the presynaptic neuron, and here we have a dendrite from the postsynaptic neuron. And so we have signal passed from an axon to a dendrite through a gap, which we call the synaptic cleft. So the neurons, in this case, don't touch each other, but rather they come very close to each other in this socket-like connection, and they message using a neurotransmitter, which is released from the axon terminal and is received by the dendrite. And there are many types of neurotransmitters, and they have different regulatory mechanics. And it's one synapse can potentially use a different multitude of transmitters, enrich the amount of communication or activity that might be happening.
Just super quick before we move on from here, are there any other questions right now? Because this is sort of like a new concept over here for some people. Okay, cool.
Yeah, feel free to just shout out or speak up. I'm totally okay with getting paused. If we don't have any questions,
Yeah.
I'll continue through the presentation, and then maybe we can have a larger Q&A.
Yeah.
And if anyone has follow-ups. All right, so the basic properties of excitatory neurons are that they have dendrites and axons, which is something we've seen in this previous picture. This is a little bit of a zoom in, and this is more of a high-level version. So also the neuron has a soma, which is the cell body of the neuron. And a neuron is connected to other neurons, again, from the axon to the dendrite, which we would see here. Axon terminal connects to a dendrite of another neuron. And there's the synaptic cleft with neurotransmitters getting passed through. And when this neuron in the middle receives a neurotransmitter from its neighboring neuron that it is connected to, it can get excited. And the excitation causes the neuron to send an action potential down its axon, which could then trigger some neurotransmitter release at its synapse and cause the downstream neuron to fire as well. And that allows us to do signal passing through a neural circuit and also allows us to do computation on that signal based on the responses of the neurons and the thresholds of the amounts of neurotransmitter it takes for the neuron, a downstream neuron, to get excited. This is just another way of viewing the same concept. This is, again, the presynaptic neuron. This is the postsynaptic neuron. And we have a membrane protein inside of the neuron, which is blocking the entrance or exits of different ions. And as we saw earlier in the in the spike activity waveform, we had a resting state, which was negative, I think negative 70 millivolts and microvolts, sorry, negative 70 microvolts, which is where the neuron likes to be at. And so there are pumps which pump out positive ions and try to leave the inside of the cell as a negative resting state. But when the membrane gets excited by, say, a neurotransmitter, then it will open and it will allow positive ions to infiltrate the inside of the cell, causing the signal, the voltage, to spike up and eventually go back down as the cell goes back to its resting state. And the brain has many kinds of these connections, and a lot of them can be very close to each other, but can also span very long distances, and that allows for communication in between brain regions and synthesis of information across the brain. And as you can see here, the density of cells and connections in the brain is quite big. But even though this looks very convoluted and tangled up, there's a lot of organization and meeting in the positions of different cells. They all play a specific role. And besides neurons, which you might be able to identify here, I'm not sure which colors the neurons are, but besides neurons, there are also lots of supportive cells, which we call glia. There are macroglia and microglia. But overall, the glia surround the neurons and fixate them in place. They supply nutrients and oxygens to them. They insulate a neuron from another, but also help with myelination, which is something you can see here. Myelination allows for more efficient transport of electrons down an axon. So you get a much faster signal traveling through the axon because the electrons bounce from one sort of gap to another gap over the myelinated areas. So it's like a little speed boost as opposed to just going through the axon. So they help with insulation and also myelination, and they can destroy pathogens and remove dead neurons to clean up the brain and make sure everything is functioning properly and is healthy. So even though this might be really dense, not all of this is neurons. A lot of this are actually supportive cells. And one really interesting supportive cell is the radial glia. And it's called radial glia because you can see them growing in a radial-like pattern, providing these stems on which we can have other neurons structured along. And this allows for the formation of cortical columns, which are a very cool and interesting computational subunit within the cortex. And now that we've dived a little bit into the brain and we kind of know the structure and we know some of the neural circuitry principles, perhaps someday we can engineer interfaces that may link the human brain with computers of other brains. If we're able to decode the neural coding of brain signals through cortical columns or other interactions, long-range interactions, and engineer better interfaces that can either go into the brain or work outside the brain. And the cool part is once we have that, diving into the software aspect of brain-computer interfacing and thinking about things like having artificial neural networks connected to real neural networks and the possibility of those two organically working together, or perhaps neural networks might have some biological or algorithmically inspired upgrades that would make them more easily compatible with the way neurons work in the human brain. And these are just some sci-fi plausible scenarios that might happen. This is something called a neural dust particle, which was developed, I think, at Berkeley. And they're quite small, and they're able to be injected in the bloodstream, and they could travel either into the bloodstream or into the brain, and they could disperse and be able to sense different regions inside the skull and transmit signals back to a receiver that is sitting outside the brain. And that might allow us to non-invasively or less invasively have electrodes or other forms that are inside the brain. And it's been predicted that direct communication between brains and computers would be a major evolutionary advance for humans. And there are several companies like Elon Musk's Neuralink that were founded on this idea. So I really like this graphic where in history, there are several isolated brains. And later on, we developed things like speech and communication, either body language or other forms, writing, that allowed us to have indirect brain communication. And in the future, we might have direct brain communication. And right now is this really interesting sweet spot where we might see a rapid advancement in transitioning between the area of indirect brain communication and direct brain communication through initiatives such as Neurotechnology, brain research, deeper understanding of the brain, and the ability of mapping and understanding how the brain is wired and how potentially we can use that knowledge to create better brain-computer interfaces. So thank you so much.
Awesome.
Are there any questions?
Yeah, about hardware, neuroscience, hardware plus neuroscience, maybe. Okay.
This
Cool.
lecture was kind of all over the place.
Yeah.
But the main areas are, I think, in relation to the club projects and club goals for this quarter, we would mostly be looking at this pipeline of how we can take brain activity, extract useful features from it using things like machine learning and principal component analysis and other analytical tools, and be able to detect features that would be useful for some kind of action.
Yeah, exactly. This is sort of the pipeline that we see like all over brain-computer interfacing. And yeah, this time we were really focusing more on like the left side of this, like between the brain, like the subject, and the hardware. But we'll actually be moving more towards feature extraction and like machine learning next week. So
yeah, and the further we can get with having solid hardware, the kind of latter part, which is machine learning and being able to have some processing, meaningful processing and input to the user could generate some interesting research
Yeah.
areas for the club, or maybe for people that are looking for grad school or industry type research.
Exactly. Yeah. Any other questions about like neurotech club stuff in general, or even research? Because Kate probably has a lot of expertise from working in like as a grad student. So any questions about that as well? Okay, cool. Yeah, Kate, do you have anything else?
So yeah, this week was neuroscience, and next week will be coding in neurotech, which will go over some of the popular libraries and approaches for trying to analyze the neural signals.
Yeah.
And if you guys have any questions, I think we'll post the slide deck maybe in Slack, or probably Slack is the best option, I'm thinking. And if anyone has questions or wants to chat about any of these aspects or some of the sci-fi stuff, then I'm also available on Slack and definitely down to have a chat after.
Is anyone here not on the Slack group right now? If you aren't, just let me know and I'll send you the invite link. Okay, cool. All right, I think that's about it then.
Thanks, everyone.
Cool, thanks. Hope to see you guys next week.
All right, thank you.
Thank you.
Bye.